This presentation aims to teach grade 12 students the theory behind enzyme function and the interesting and innovative applications of enzymes in various fields.
My name is Patrick Hafenberg, and my partner's name is Tristan Grovu. This is our presentation on enzymes and their applications.
So, what are enzymes? Enzymes are types of proteins that react with substrates and have the ability to catalyze or increase the rate of these chemical reactions. A substrate is simply a type of molecule present in a biological organism that needs to be converted into a different molecule so that it may be used. In a laboratory setting, these types of reactions would require conditions not possible in the human body, including different pH levels or pressures, as human bodies require a very delicate balance. That's where enzymes come into play. They allow these reactions to proceed without drastically changing any biological conditions. Enzymes are usually identifiable by their names since they often end with the term "ase".
This illustration depicts the way enzymes catalyze biochemical reactions, which is by lowering the reaction activation energy. Activation energy is the amount of energy needed to start a chemical reaction. If this energy threshold is too high, it will be very difficult for the reaction to proceed and little product will be created. So catalysts, in this case enzymes, perform intermediate reactions that have lower activation energies than the initial reaction, allowing the overall reaction to easily move forward. However, since enzymes are intermediates, they are not actually consumed in the reaction. Enzymatic reactions can be broken down into four major steps. Firstly, a substrate with the complementary receptors binds to the enzyme at what is called the enzyme's active site. This binding depends on both the chemical reactivity and shape of the substrate with respect to the enzyme, which is called molecular recognition. This then produces an enzyme-substrate complex, which undergoes chemical reactions and converts the substrate into its corresponding products. Once the reactions are complete, the products are released from the enzyme, and the enzyme is now ready to bind with another substrate and repeat this cycle. The lock and key model describes the method by which substrates bind to enzymes. It was developed at the end of the 19th century by German chemist Emil Fischer, who theorized that substrates connect to enzymes in a similar manner to how a key fits into a lock. They have preset complementary shapes that fit exactly into each other. This was a good model as it provided an explanation for enzyme specificity, but it did not consider the state between substrate and product, known as a transition state, and was thus considered an incomplete theory.
Another theory explaining how substrates bind to enzymes came approximately 60 years later and was developed by an American biochemist of the name Daniel Koshland. Koshland's theory is similar to that of Fischer's, as the proper shape and reactivity of the substrate are still required to initiate binding to the enzyme, but it differs as the enzyme's active sites are now believed to adapt their shape to better hold the substrates. Thus, active sites are now believed to be flexible rather than rigid.
Some enzymes require additional molecules called coenzymes or cofactors to properly activate and bind to a substrate or catalyze a reaction. Cofactors are usually a metal ion, and coenzymes are other small organic molecules.
The following are certain factors that influence how enzymes function and include pH level, temperature, activators, salinity, substrate concentration, enzyme concentration, and inhibitors.
Changes to the pH means changing the hydrogen ion concentration, and if the changes are drastic, the enzyme characteristic properties are destroyed and the enzyme is denatured, and this also results in changes of the enzyme's 3D shape. Thus, it has been determined that the optimal pH for most human enzymes is within the range of pH 6 to pH 8. This figure shows the change in enzyme activity as pH becomes more basic or acidic, and it is evident that the different enzymes have different responses depending on their properties.
Greatly increasing temperature has a similar effect to drastically changing pH levels as the enzymes denature, decreasing enzyme activity. This is due to the increase in thermal energy, which in turn causes the weaker bonds within the enzyme molecule to break, thus converting the enzyme into a different molecule. Decreasing temperature also decreases enzymatic activity, since lower moving molecules leads to less collisions and thus less interactions between enzymes and their respective substrates. As such, the optimal temperature for human enzymes is determined to be between 35 and 40 degrees Celsius.
Modifying salinity is very similar to changing pH level, as both influence ion concentration. This affects the structure and the bonding of the enzyme molecule, causing it to change and denature. A good example of this is the Dead Sea, where only select life forms exist due to certain enzymes only working in certain salt concentrations.
Just like how some enzymes need cofactors or coenzymes, other enzymes need allosteric activators. These are molecules that bind to an enzyme, effectively changing the shape of the enzyme's active site so that it can better bind and hold on to a substrate. However, these enzymes are dependent on their respective activators, and as such, if only a few activators are available, then the corresponding enzymes will not function effectively.
Enzyme activity is proportional to enzyme concentration, as well as substrate concentration, since an increase in enzymes or substrates would increase the collision rate between the two, thus increasing enzymatic activity. If both concentration increase at the same time, then there would be an increasing linear relationship between concentration and enzyme-substrate reactivity. However, if only the enzyme concentration increases, while the substrate concentration remains the same, the concentration reactivity relationship is as seen in this figure. It will eventually plateau, and no matter how many more enzymes are added, the reaction rate remains the same. This is due to the substrate concentration acting as a limiting factor since it remains constant.
Enzyme-substrate reactions may get blocked by molecules called inhibitors. These inhibitor molecules bind to either the active site or the allosteric site and decrease enzyme-substrate reaction rate. Inhibitors can be classified as either competitive or not competitive. Competitive inhibitors bind to the active site of the enzyme, preventing the correct substrate from interacting with the enzyme, and thus the correct substrate must compete with this inhibitor. Non-competitive inhibitors bind to the allosteric site of the enzyme, which then changes the shape of the active site and prevents the enzyme from interacting with its corresponding substrate.
Some types of inhibitors are also able to permanently shut down an enzyme's reactivity by covalently bonding to functional groups at either the active site or allosteric site. This bonding means the inhibition is irreversible and substrates are no longer able to interact with this enzyme.
Another type of inhibition is feedback inhibition. This occurs when there is a chain or cycle of enzymatic reactions where one reaction allosterically inhibits a previous reaction. If one of the participating reactions is slower than the others, in this case reaction D, then it acts as an inhibitor as it slows down the entire cycle if the product from one reaction is needed as a substrate for a previous reaction, in this case reaction A. This is because the slow reactions do not produce the fuel for the next reaction at the same rate that the fuel is used, which causes the next reactions to slow down, as well as the reaction after that, and so on and so forth, until all reactions become dependent on the slow reaction.
Lactose intolerance is an issue that affects the majority of the world, typically affecting people of East Asia and West Africa with other regions. Overall, a decrease in lactose digestion ability is found among 65% of the world. Lactose intolerance occurs when lactose is unable to be digested due to the lack of lactase enzymes. Lactase breaks down the sugar lactose into glucose and galactose as shown in the picture. Lactose pills may be taken by people that suffer from lactose intolerance as it increases the lactase enzyme count in the body, allowing the molecule lactose to be broken down.
A recent breakthrough has been made in regards to plastic recycling. Petroleum terephthalate, the plastic making water bottles and soft plastics, can now be infinitely recycled. A company called Carbios has come up with a solution to use enzymes to digest petroleum terephthalate, or PET, by depolymerizing it into its separate monomers. Once this is complete, the monomers are purified and the cycle is repeated with different enzymes. This allows PET plastics to be reused almost indefinitely, which is amazing to reduce waste.
We hope you were able to take away the theory behind enzyme function and the interesting applications of enzymes in different fields. Feel free to contact us at the following emails if any questions arise. Thank you for listening.